So I think, oh sorry, I just missed. You can hear me also. Can you hear me? Yeah. So can you hear me? Yes. So yes has come for that you are able to hear me, correct? So do I wait for 2 more minutes? Please make sure that everybody is here. I don't want to repeat it, and I need to finish this today. That becomes the last one for this academic semester. Right? Good evening there. So you people could know which one is our DSE for next semester. If you could make, have you read? Anybody can write, which I had written at some places, which is our DSE for next semester. Anybody is knowing that? DSE for the next semester is which one? Anybody knows that? Does anybody know which one is our DSE in botany for next semester? I have a file with me, I can just tell you. Just give me 1 minute. In the meantime, all of you join. By chance I have left that in the college. Maybe he shared with you. So I start with this today's class today now, and we'll do rest of the things later. So everybody can hear me and everybody can see me, right? Right. So I'm starting with this, and you know what we'll be studying today is variation in chromosome number and structure. And I tell you, if you study genetics and if you don't study this topic, your knowledge will always be incomplete. And this becomes the most important segment of, a very important segment, if not most important segment of studying the genetics and molecular biology. You studied Mendelism, you studied many things. Just give me 1 second. Right. So you have studied Mendelism, you have studied cytogenetics, partly the post-Mendelian era, linkage, crossing over, but that becomes this is equally important topic, and we start with this. And once you study this, then you will know how important it is to study the chromosome number and structure. Variations, meaning there is one normal thing and there is a varied thing, right? So do you see this picture? Can you, I hope you all are seeing this picture which is there. on your screen. And what does this picture show you? It shows you what? Like what I want you to notice at this structure or revise in your mind is that it shows you the chromosomes, and the chromosomes are arranged in a specific pattern that we call as what? That we call as karyotype. So you all know what is a karyotype, but still when I am teaching you chromosome number variation and structure variation, I will be showing you this karyotype again and again. The two things which are important in this karyotype is, one is, it's done, and I am less interested in your attendance, and I am more interested in teaching you this topic, which is very important, and I want to teach you in a way that you have learnt it. And like we recorded this, why? Because if you have learnt it and still if you feel that you missed some part of it, you can go back and revise that what I was telling you there, right? Okay, so what is a karyotype? Two things important in this karyotype here is that you have all the chromosomes of a organism or a human being, and the other thing is that the chromosomes, different regions of the chromosomes have been stained differently, correct? So these are the two. They are arranged they can be decided to be arranged in any manner, but their different regions are stained differently. And secondly, each pair of chromosome is given a number, right? So if it is for human beings, it will have 23 pairs of chromosome. So this is what we are studying, and I started with this, and now I tell you what all you will be able to learn when you study, or after you have studied this thoroughly. What all you would have learned from this class is that you will know what is karyotype and how important it is to know and make a karyotype and understand what are the implications of changes in the karyotype. And then we want to know what is the, can any one of you write what is the difference between karyotype and ideogram? Karyotype and ideogram. See, this is how I know that you are actually listening, and my energy is not going waste, that the students are listening, and you can find out the answer, and I expect for this answer in 1 minute. Is what I want from you is what is the difference between karyotype and ideogram, and you have to write the precise answer, please. And I am waiting for, I see you have 45 students in your class, but I don't see more than 8 here in the class. So for those 8, at least they should learn it well, and you should be interactive, otherwise I'll stop this class, right? So first thing which I asked you is what is the difference between ideogram and a karyotype, right? You can start writing. In the meantime, I'll tell you other things. I'll be teaching you number changes, that is haploidy, monoploidy, yes, is a type of karyotype, and what is the difference? The number changes, haploidy, monoploidy, euploidy, polyploidy, we'll talk about that, and we'll touch upon what is the application of polyploidy. We talk about several structural changes in the chromosomes, and we talk about transposable elements, repeat elements, duplications, deletions, translocations, bridges and breakage, fusion cycle, which I find very interesting, and you will also enjoy once you know what is that. And then chromosome bridges and laggards. Little bit about that, because I learned that in the practical the students were lost, and they didn't know actually what is meant by this. I understand you people have a lot of pressure of other subjects also. Yes, is the actual picture of the chromosome is fine, but you tell me the exact difference. Exact difference is karyotype, ideogram, of course you said is a type of karyotype, but in karyotype we have different regions stained, first of all, by differential staining, and secondly we can take any criteria to arrange them, and the third thing is we label them by number 1, 2, 3, 4, 5 in the karyotype. Yes, she said right. And in ideogram they are arranged in decreasing order, and we don't bother about staining in ideogram, right? And then let's go further, and I start with this. What is this? Yes, correct. Absolutely. So let's start with this. Here I am showing you different things and how study of karyotype can be important to you is that the karyotype can change due to rare events, like mutations are point meaning small changes, but if you have changes in the chromosome, the karyotype will change and the changes are inherited. Those changes are inherited and it's important to study the karyotype and to observe the changes. And you know, the changes, the extent of changes can vary within the species and the karyotype evolves with time. One interesting example which I could find for you is of this barking deer, which has several species across the world. One interesting species is Chinese munzak, and we have Indian munzak. And you know what happens? In Chinese munzak, you have chromosome number 2n is equal to 46. So I hope all of you understand the difference between 2n and 2x, right? What is the difference between these two? I would love to hear from you. When you write a genetic formula, what is the difference between these two, right? And so I said in Chinese barking deer we have 2n equal to 46, but in Indian barking deer the chromosome number varies. It is 2n equal to 6 in females and is equal to 7 in males. What does that mean? It is 2n equal to 6 in females and 2n equal to 7, right? So they still can be maintaining the same characters that are of a species, but their chromosome number are so different. Can you believe it? That is 2n is equal to 46 and here the 2n is equal to 6 in case of females and 7 in case of males. So things are so different, but the species is basic fundamental species remains the same. So karyotype evolves and karyotype can be different, can have different a number, and it varies and the changes can be huge. And one more example which I could get you for you is that you see a very small change in chimpanzee and humans is that in this chromosome just the sequence is inverted. So it was just a very small change in the chromosome, that one sequence was inverted and that led to what? Changes in the species, that the species now are different. So with these few examples I tried to show you what? I tried to tell you that the chromosome number is chromosome structure, number, karyotype study is very important and it can lead to different conclusions, and the species, their karyotype may be very different, but they look similar. And you have to remember both the examples, one for inversion I gave you and the other one was for barking deer. The Chinese species and the Indian species is so different. Then let's go further, is variation in. So this was just an introduction, I was telling you that how do we study the variation in chromosome number and what is the implication, and then we were studying that the changes can be huge or they can be a very small change in the sequence and the species become different. There was one case where a huge difference in the chromosome number, but still these species were same, and in another one there was a small inversion but the species were different, right? You can still, Jyoti, ask any question whatever you want to, you can stop me wherever you want to, but the point is that in your first year you should not be weak in these topics on the basis of which we are going to teach you further in the next coming years. They become their fundamental things, right? So let's go further. Now I am talking about variation in chromosome number because I told you I'll teach you all this in 2 heads, right? So variation in chromosome number is what? It can be whole set of chromosomes involved in variation, that we call as euploidy. But if a partial set, 1 or 2 chromosomes are involved in variation, that we call as aneuploidy, right? So we have euploidy if the complete set, aneuploidy if not complete set but part of the chromosome number, right. So euploidy is multiples of the one complete set that we call as X, right. So in haploid you just have one X and you have N, and in diploid you will have two set. So if we take the example of ABC here, in haploid you will have just one set of ABC, in diploid you have how many sets? Two, and in polyploid you have, if it is triploid you will have three, and if you have tetraploid you will have four. And in case of, let's talk about aneuploid, if it is monosomic you have one missing, like C is missing here, that becomes monosomic, and in trisomic you have one extra, 2n plus 1. So this becomes a convention to write these things. So how many of you remember meiosis well? Because in the last class also I was discussing about meiosis. That is the fundamental of life. When you study mitosis, meiosis, you should understand its implication also very well. And I hope you remember the step where in the meiosis from metaphase, first in the mitosis from metaphase, when it reaches to anaphase, what happens? The centromere divides, and when the centromere divides, what happens? From one chromosome we have two chromosomes, right. And in meiosis on the other hand, the two chromosomes were homologous chromosomes, they separate in anaphase, but they are still connected by what? The crossing over or the chiasmata point. So how many of you agree with me here? I am just seeing students are there, but whether they are actually, I want it to be interactive. So how many of you agree with me that in meiosis the homologous chromosomes are still attached by chiasmata? And I hope you know what is chiasmata. Chali, I am waiting for your answers. How many of you agree with me that in meiosis there is no centromere dividing and the chromosomes are still connected by chiasmata when they start separating for in anaphase, anaphase 1 especially? Right? So Jyoti, are you trying to write the answer for this? What is chiasmata? Can you write what is chiasmata? See, when I was interacting with you in your Viva, I found some of the students to be very, very promising. So if you actually want to make a career, yes, correct. In the metaphase 1 of meiosis, they are connected with chiasmata and not in metaphase 2. So that gives me a good feeling that I am not speaking in front of wall and the students are listening to me and are attending. And I tell you one more thing here, that the hard work never goes waste. Just keep this in mind, you are not wasting your time, you are using your time. You never know which thing is there in the question paper in the examination. And again, the idea of cramming up things is not going to help you. So I hope by now you have known that you remember while karyotype will be used again and again, and we are talking about variation in chromosome number, and now we have known the euploidy and aneuploidy. In aneuploidy there will be changes in one or two or few chromosomes but not the complete set, but in euploidy will have a changes in the addition or deletion of the complete set. So aneuploidy, you are also students of zoology along with botany, that aneuploidy is very, very important, and because it leads to several diseases and the phenotype, this genetic disorder leads to changes in the phenotype, and the typical phenotypic changes or the example of the genetic defects usually are slated eyes, maybe slanted eyes, rounded face, cleft palate. So it could be anything, and you have studied already about Down syndrome and Turner syndrome. So aneuploidy is important because in humans it is causing several diseases. meaning not diseases with several abnormalities which many of which could not be survived. They are lethal, but some of the individual survives, like Turner syndrome and Klinefelter syndrome. So, but aneuploidy is very, very important. Now let's come to polyploidy. Now, why polyploidy is important? Polyploidy, we have triploid, we have tetraploid, we have hexaploid, we have octaploid, and many more, right? And again, I tell you one more very fundamental things when studying polyploidy is again this reduction division, or in the mitosis when the centromere is dividing, right? Because say for example an organism is triploid, right? In triploid, what will happen in the metaphase? You will have, in the meiosis, you will have what? 3 chromosomes instead of 2 arranged or making the synapses. You agree with me on this or not? In case of a triploid, yes. X is the basic, yes please Sujal, yes, correct, very good question. And I tell you, what is this? N is the one set of chromosome in any organism. Like I tell you the example, any example you can take, maybe sugarcane. Sugarcane is 6x, right? X is the basic chromosome number, so is 6x, but 2n is, say for example, it's 6x is 48, right? So x becomes what? 6, right? But if we write 2n, we'll say 2n is equal to 24. Got my point? X is the basic set of chromosome number, and 2n is the, when we say that the diploid number is this, because in some cases it's very, very different and things become different. Why? Because in the polyploids, where we'll be talking about just now, you will see what, that especially the autopolyploids or the polyploids, the intrageneric or the amphipolyploids, what happens is that they have obviously come from different organisms, but they behave normal. Now their 2n become like 46, I said for sugarcane, or maybe but the x is 6. That was a hypothetical example, but I hope you could understand what I am saying here. Got it? Or still you want to discuss it further? I'll be very happy if you want to discuss it further. Or let's go further. You will see that this thing is coming again and again in this topic, right? So polyploidy, you all know what is polyploidy, and we have more than 2 sets of the chromosome, right? Yeah, we have more than 2 sets of the chromosome, and it can be autopolyploidy, it can be allopolyploidy, it can be amphipolyploidy. You know this, that the polyploidy is very, very important in evolution, and you also know that the polyploidy is larger in size. So what happens is that the polyploid organism is giving you more food or more output, whatever you want in terms of amount. So polyploidy is very, very important for plant evolution, meaning new species arise very often, and polyploidy is also very important in plant breeding, because the several examples of cultivated potato, for example, potato which we eat is a tetraploid. Again I am at your question, Sujal, again, right? So potato is what? Potato 2n, if you write, you will say it is 48, but basically it has 4 set of same chromosomes, so it will be 4x. And why you should know this? Can you guess, Sujal? Why should you know this, that what is x and what is n? Try guessing, because I told you just now, I was coming to that example. Say it is, if it is triploid, right? Like again in this solanum, in meiosis what will happen? In meiosis they will arrange, there will be set of, there will be, meaning diploids, it will not be bivalent there, it will be 4 chromosomes arranging together in the metaphase plate for separation. You got it? and that need to be studied and taken care of very carefully, because basic chromosome set is 4, but the nature has evolved in its way. And same way, if you see in the sugarcane also, sugarcane is octoploid, the 2n chromosome number is 80 and it's 8x, so fundamental chromosome number is 10. Alright with this, I hope you could get it, and I said it is very very important. Still, when it is an even number, normal meiosis can happen. But if the number is odd, for example, triploid I was talking to you, pentaploid, do you ever hear a pentaploid being successful in nature, triploid being? Because it's hard. Why? Because no meiosis, normal meiosis can take place, because when they separate during meiosis 1, because it will always be uneven. You agree with me there? So I told you what is autopolyploid. Autopolyploid, when we have same basic number, meaning auto, it is coming from the same, but it is represented several times. And in the I gave you 2 examples here, that is of sugarcane and potato. Now, what is allopolyploid? Allopolyploid is not coming from one genus or one species, meaning what happens here is we have different organisms, different species contributing, not to the genus, different species in nature, they have contributed, and they have evolved gradually with the gradually to make a new polyploid. Say, if you want to see the example of allotetraploid, that is our bread wheat, if you see the 2n is equal to 14, meaning one basic set of chromosome which is coming from different organisms is 14 different species, right? And the total is 42 because it is a tetraploid. So it is coming from icon wheat, and one set of chromosome is coming from grasses, and then it together finally our bread wheat is a tetraploid with chromosome number being 42, and the wheat is also hexaploid very often, so it varies from species to species. But the difference in auto and allo, I hope you could understand, is in the allo we have different species involved. Then we have amphipolyploid. What is amphipolyploid? Where we have different genus involved, and you know the different the 2 genomes derived from different species, different genus. And in, like, you have 2 examples here is triticales is man-made cereal, this is what we call it. And what happens here is that is a cross between rye and wheat. Triticale, why? Because secale cereal and wheat is triticum. I just add a picture of here, and this, if you look at the ploidy level of triticales, it has 4 ploidy level, and it's a decaploid basically. It may be octaploid, it may be hexaploid, it may be tetraploid. So you can define accordingly depending upon the basic set of chromosomes which you have here. So I hope you will be able to write the differences between allopolyploid, autopolyploid and amphipolyploid, right? And the difference is huge in amphipolyploid, and you get a new, absolutely new species, right? Is that okay with everyone? Aneuploid, polyploidy, euploidy, autopolyploid. देखिए आपको सब ये terms, I hope you could get all of you these terms, right? So let's go again. The important thing which I was talking just now, because that's the most important thing which I feel you must understand, is that what is going to happen in the meiosis, right? The errors come from where? In the nature. I showed you the wheat bread wheat, how it evolved from two different species, it took chromosomes from different places. But how this happens in nature is that sometimes during meiosis, what happens is that there is non-disjunction of chromosomes. Say for example, this AABB here, it might as well happen that all the chromosome goes to one egg and that egg is fertilized, and the other cell did not have, meaning it didn't miss, there was non-disjunction, they could not separate during meiosis. It might have happened with all the chromosomes, or it might happen with one chromosome. And what will happen if it happened with one? It will be trisomic, and the one where that chromosome was missing, it will be monosomic. So what is happening is, we can discuss this aspect from the point of view of complete set of chromosomes and one chromosome also. But here in this picture we have shown you one chromosome. So in non-disjunction it leads to aneuploidy, and what happens here is during the separation, during the gamete formation, if this chromosome, if this is AB, what happens is both of them go to one parent and one gamete, and the other one is without it. So if they get fertilized, what happens is you will get trisomy or monosomy, meaning it might be absent or you might have one extra copy. So this step which I was talking to you is very, very important in meiosis and in aneuploidy or polyploidy, because we sometimes induce polyploidy. Why? Because no spindle fiber formation, so they cannot separate, that leads to polyploidy, right? And here in aneuploidy it happens only for one chromosome. I hope you remember this picture always in your mind, and this will help you in understanding. And the term for this is non-disjunction of chromosomes, that they are not able to separate during meiosis 1. Okay with this? And I said this is a very important picture which, while studying the variation in chromosome number, which you must keep in your mind, right? So, and I told you this already, this variation in chromosomes is a big cause of abnormalities in the gene expression, and you get a different phenotype if there is aneuploidy. And I told you the reason. The reason again, because when it, first of all, is that basically our genome has evolved in such a way, is that if there is aneuploidy, it results in unbalanced genome. If the genome is not balanced, there will be abnormal development, and this abnormal development leads to what? To the death of the organism. So aneuploidy usually leads to the death of the organism, but embryo with one or few chromosomes, with one extra or one too few chromosomes, has, of course it has unbalanced genome, but they survive. I tell you again, the aneuploidy is very, very common, and it occurs normally in 3.5% of all the embryos. And aneuploidy for large chromosomes in humans usually causes unbalance and usually is lethal. But if the chromosome size is small, and again, you know, it is lethal and it leads to stillbirth, and 20% of the birth are still because of this abnormality. But if it is in small chromosome, it may be viable, like Down syndrome is for 21 chromosome. It comes because it may be that the complete 21 chromosome has come, or even if a part of chromosome has come, it will lead to Down's syndrome. And why Down syndrome happen is because of the non-disjunction, which I explained you just now, in the females during meiosis 1 or meiosis 2, right? And sometimes it also happens because of non-disjunction in males. In 25% of the cases, it might as well happen because of non-disjunction in males. So I gave you this example of trisomy, and I told you it is so important in human beings, and it leads to unbalanced genome, and it is the cause of up to 20% of the stillbirths. So all of you agree with me there? All of you agree? You could understand the difference between euploidy, aneuploidy, and the examples for aneuploidy and the examples for polyploidy, and what is the most important thing there? Yes or no? Chali, I am waiting for your reply. हो गया? Yes ma'am, yes ma'am is coming. So I get a feeling that the students are listening to me. Correct. So let's go further. Thank you. So I have done chromosome number variation, and the important terms and examples I told you, it might come in any form in the exam, but you should be able to write about this now. Aneuploidy, the different types of syndromes that will come in the exam, in the practical also, and polyploidy, applications and examples, auto, allo and amphi polyploidy. So I think you should be able to write it there. So now let's reach to variation in chromosome structure. So variation in chromosome structure, and if you remember the very first picture I told you, in the very first picture, what was that? In the very first picture we had made karyotype. We had made a karyotype, and in the karyotype, you know, it was showing bands of different types. So if there is any change in the chromosome structure, you will get a different band. And I hope you remember the very first karyotype where I had made a inversion in the chimpanzee and human beings. A small inversion has led to such big changes. So in studying the chromosome structure variations, we'll be talking about the variation, the karyotype again and again. And I hope all of you know transposable elements. I am telling you 6-7 things here in variation in chromosome number, chromosome structure, and what happens, why these chromosome structure variations come. And if you still have any questions, you can ask me from variation in chromosome number. I am continuing, but I am consistently looking at the comments from your side if you want to. And I really liked that 2n and 2x, it often comes in the exam. So the first thing is transposable elements or jumping genes. All of you know what are they. What are transposable elements? If you want to write the definition, they do what? They keep moving in the organism from one place to another, right? They are also called as jumping genes. It's okay, they keep moving from one place to another, and they might move as it is, and sometimes they duplicate and one copy moves. It's okay that they keep moving, but what happens? Wherever they go and they insert, they change the sequence of genes or they change the chromosome structure in that place if the change is big. So this transposition can be of two types. One is duplicative. So one portion, if you can see in the figure, one portion duplicates and it goes and attaches to the another region, and non-duplicated is that portion itself jumps and goes to the new place, but it leads to big changes in the chromosome structures, right? And this can be, they can spread anywhere, and sometimes that these changes are not very good, and sometimes because of this but my idea is not telling you about the transposable elements here. My idea is to keep my focus on the variation in chromosome structure, and I hope you will agree with me here is that the transposable elements can change the genome structure or the chromosome structure because they move from one place to another, and the sequence of genes there change because something new has come and has added itself or has inserted itself there. So you must know the definitions and about the transposable elements. You know, Barbara McClintock, she discovered these transposable elements because it was a mystery before that how come suddenly, you know, the structure of the genome changes without any other thing has happened in the cell. So this transposable elements was a big discovery, and very often these transposable elements are causing big changes in the chromosome structure. right? And you should know one example which I have drawn earlier. You can write this example if it comes in the exam, to write the role of transposable elements in change in the chromosome structure. Similarly, let's come to the repeats or repeated sequences. What happens in there is, what happens in the repeat sequences are what? All of you know what are palindromes or repeat sequences, palindromes. All of you know what are palindromes. Who want to write the answer, please? I am waiting. What are palindromes? Palindromes, the changes, palindromes are what? I want a definition of palindromes from you. What are palindromes? देख के बता दीजिए. You can find it out from Google. Yes, correct, which can be read from both the sides. But what is their implication in variation in chromosome structures? Is that they sometimes arise in nature? Yes, Harshita, yes, correct. And what happens is that they arise by replication slippage, meaning extra base is added or there is a change in during replication, these sequences, but they causes big changes in the chromosome. Yes, correct, Khaneja, Sujal Khaneja, right. And sometimes long repeated sequences also arise, and that is because of this duplicated transpositions, is that they are coming from one place and getting inserted at the other place, and they lead to changes in the chromosome structure. So that was these two things, though they were not directly covered in the variation in chromosome structure, but still they are important, and they are important to know. But let's come to the main thing, that is, first here is duplication. What is duplication? Is that the set of genes, they got duplicated, right? They become in more than one copy. Say for example 1 2 3 4 5 6, because of duplication of 4 5, it becomes 1 2 3 4 5 4 5 6, right? So duplication can be because of any reason. Mostly it is because of, again, the duplicative transposition, that the one portion is moving and getting inserted at the other region, or maybe the similar region. It causes the duplication, and it is sometimes, because of the it gets duplicated and gets inserted in the pseudo genes, and those genes become active and they are representing the duplicated regions. Now, who is explaining me what is pseudo genes? I am waiting for your answers. What is a pseudo gene? Pseudo gene. I am talking to you about pseudo genes. I am waiting for the definition of pseudo genes. Yes, that's okay. Now I asked you about pseudo gene to an inactive form. So they are like gene, but they are inactive. Correct. So what happens is that in duplication is the pseudo genes are correct. Absolutely. So pseudo genes, though they were inactive, but because of transposition, what happens is that the duplication that goes and gets inserted in the pseudo gene, the gene suddenly becomes active. So this leads to big changes. It's a simple example of duplication, but it leads to big changes. And so transposable elements is one reason, and the other one is that duplication can occur because of the unequal crossing over. Very often the crossing over is not equal, and if we have the synapses, during synapses if the sequence is inverted, so they don't exactly synapse, sometimes they do not exactly synapse and the crossing over is unequal in that situation. I have a diagram in the next slide, I will be able to show you that how this synapses and especially the inverted repeats, they cause the unequal crossing over. So one chromosome gets more of the sequences and the other get the less. And a typical example, we talked about this example in very much details when we were doing what? We were doing complementation and gene mapping. The same example had come there, and we were talking about deletions and how the duplication is leading to the double bar or wild type character. Wild type is normal I, but the double bar is if the genes get duplicated, it leads to the double bar I in Drosophila. So duplication is happening routinely and normally also, and transposition and crossing over, unequal crossing over are the two main regions for this duplication, right? And I told you again, I was just telling you this, that the duplication is very, very important in evolution, because if we have instead of one genome, one sequence or one chromosome, if we have two set of that, what will happen? If one set changes, we are not changing, but that set will change to a new thing. So we acquire a new character. You agree with me there that the diploids have two genomes, but if it is duplicated, we will have three sets. So one set always has the possibility of changing without actually changing the organism, so it becomes more adaptable. So, right, because of duplication, if this AA is represented 3 times, so one A can always be either it is adding to the junk DNA, non-functional, it becomes non-functional, or it evolves to a different to a recessive, or maybe to a different one from A dash to B, entirely different. So when it is neo-functionalization means what? A new function, it has acquired a new function. So duplication is very, very important in evolution, right? They are very important in evolution. And the other important thing about duplications is, in duplications usually they might add to the junk DNA, I told you, and usually the genes they evolve to are quite similar. Say for example, this myoglobin protein gene has evolved from hemoglobin gene duplication. So usually they acquire different function but very similar function. So duplication is very important. I gave you one example. Myoglobin has evolved from hemoglobin gene basically by the fundamental principle of duplication. So that was about duplication. But all did I tell you in duplications? Duplications happen in nature, and the two cytological reasons for this, one is the unequal crossing over, and the other one is what? And the other one is that transposable elements, they are duplicative, they replicate and add to a new place. And the duplication is important in evolution, and I told you with the example of hemoglobin and myoglobin that they are usually related, that they evolve to a new function and they are very often related in their function later on. So that was about duplication. Then I come to deletion. Deletion is what? Again a loss of sequence of a region of the chromosome, and it also happens during crossing over, as I told you last time, is that unequal crossing over. One chromosome gets the two set of the things and the other one gets what? Nothing, right? And I was telling you how in the repetitive sequences it can happen very easily. If you see this figure, what happens is that if they are similar sequences, they will make this loop like structure to match with each other during meiosis, and then because of crossing over in this loop, what will happen? One chromosome will get one sequence twice and one will be without it. So basically it is in the repeat sequences. In the repeat sequences during the crossing over, they form a loop, and when the crossing over happens in this loop, the one chromosome that misses and the other one gets a duplication. And the deletion is loss of a segment of a DNA, and the result of that loss can be huge, like deletion in one chromosome led to the death of the organism. I just now told you about aneuploidy also, because genome balancing is a very important thing which has to happen. If that is not happening, it leads to the death of the organism. Same way for inversion. Inversion again arise, the way I told you, deletion is because of intrachromosomal crossing over between inverted repeats. You all told me what is palindromes, what are inverted repeats, ABA, ABA, and I hope you know that they will make a hairpin like structure when they match each other during meiosis. And what will happen is this hairpin like structure you can see, I hope, and when you have crossing over in here, it will lead to what? Inversion. It will lead to an inversion, and inversion also has an implication in phenotype. So inversions usually arise because of intrachromosomal crossing over between the inverted repeats. See, if you keep doing it like this and continue with me, what will happen is I'll gradually take you to higher subjects and we'll be able to do very good thesis in the 4th year. But that is why, you know, it takes me a lot of time, but still it is good. We'll know what all we have learned, and so it will go very good that way. So I told you deletion, duplication, transposable elements, repeated sequences, and inversion is again that happens in the inverted repeats, is a reason for chromosomal variation in the chromosome structure. Next one is that reciprocal translocation. I asked you this question in the Viva. What is the difference between reciprocal translocation and crossing over? But many of you could not answer. It's very much like the crossing over, but it happens in the non-homologous chromosome, meaning chromosome 1 can change its part with chromosome 10. So that becomes what? Reciprocal. It's not change, it's exchange. They exchange their parts, so that becomes a reciprocal translocation. And I wanted to show you this. I had added that picture. I can show you here. Right. Reciprocal translocation, and I was talking about ROHIO in the class, if you remember. What happens in the ROHIO? If you see the metaphase or early anaphase, what will you see? You see a complete ring in the center. All of you had seen this in the practical also. So what happens in ROHIO? You have 2n equal to 12. So what will happen during meiosis? They will do what? They will make 6 sets in the center, but what happens in the 6th set? There is a reciprocal translocation in all the chromosomes which has happened. Are you listening to me? It's a very interesting example which all of you must remember. I look for your yes now. So what happens in ROHIO? ROHIO discolor is that it has 6 pair of chromosomes. So during meiosis, what happens is sequentially they change their part. Please look at the figure in front of you on the screen. What happens is that, look at this chromosome. B has first chromosome. Suppose my arrow is at the first chromosome. This first chromosome has changed its part with the second one. So first B and second B is same, and C of second and third is same. This is a hypothetical example. I have shown you only 4 chromosomes. Right? And this third has changed its part with 4. Now what will happen during meiosis? They will not make bivalents as you see in the normal meiosis. Synapses will happen, but all of them will be joined together, and when in anaphase they are pulled by the centromere to the different ends, what will happen? They will still, A will like to be with A, B will exchange parts with B, but it is in different chromosome. So the net result of this is what? You get a ring like structure before they actually separate during anaphase. So this is the example for what? Reciprocal translocation. What happens is they exchange parts, but the chromosomes are non-homologous, right? So what happens is when they separate during anaphase, they break, and they are still joined by one region where they have exchanged parts, and that way in Royo they form a complete ring like structure. So you must remember this comes in the exam also. And again, I saw you had a problem with Lagarde and crossing over also, again the chromosome bridge, because the concept you should understand, then only you know what is happening. Is it alright? You could know how the Royo discolor forms the ring in the during the meiosis, and a ring, all the chromosomes are involved in the reciprocal translocation. Not one, all of them. That is why, if not all of them are involved, then you will see not all the chromosomes together in one ring, you will see small rings are being formed, 2 or 3. But in Royo discolor, this is the important thing which is happening. So now you will be able to answer the ring, the reason for the ring in the Royo discolor. It is the reciprocal translocation. अगर आप इसको एग्जाम में देखिए, I have specifically selected these examples for you guys is because you see you can write this way in your exam, and you can make a chromosome also, but still writing this makes sense. Is that what is happening is in the reciprocal translocation is they exchange their parts, and exchanged, it's not one way transfer, it is the exchange, right? You can show this in the chromosome also. And reciprocal translocation very often comes in the exam, and then I expect you to write the example of Royo discolor also. There is one important term which sometimes come, that is called as Robertsonian translocation. What is that? What is this Robertsonian translocation? Is that sometimes what happens is that is the long arm of 2 different chromosomes. Can you, all of you are looking at the figure on your screen, that the long arm of 2 chromosomes, they fuse and make a new chromosome. Small arms are lost, the long arm of 2 chromosomes, they fuse. This is also a chromosomal abnormality, and it leads to a new structure, right? And this is how, you know, if you remember the example of barking deer, the Munzat, in the Chinese and Indian, I was telling you the chromosome number of course has become less, but they carry all the information. So this Robertsonian translocation principle again is important, and you must remember that adds to your understanding also, that how much important the chromosomal structure variation could be. So I told you one example of Royo discolor for reciprocal translocation, and again the other example was Robertsonian translocation. What is that? It's a type of translocation, but it's a unique new thing happening. It's not reciprocal, as you remember. What is happening here is that the short small arms are lost, and the long branches fuse with each other to give rise to a new different chromosome. That is also a method in the variation in the chromosome structure, right? And then we get a rearrangement, and crossing over repeats, and that leads to entirely new combination, and these combinations are very, very important for evolution, right? And these rearrangements, they affect the mitosis and meiosis, and I explained you that because before going ahead in your life, in your studies, you must remember very well all the stages of mitosis, what is the difference between mitosis and meiosis, and how do you identify. See, I tell you, we were also small students like you, but what used to happen is then we understood the implication, and you will not be able to go very ahead if you don't understand the difference between theory and practical. So like I tell you one small example here. During the MSc master's time, what happens? We study the same mitosis, meiosis, same mitosis, but we take the pictures in the mitosis or meiosis, and then from that pictures we cut the chromosomes and we arrange them and make a karyotype. And now when I am studying this, if I am studying one organism in different environments, and my objective or my hypothesis is that this environment is leading to change in chromosome structure, I can actually study that. But if you are studying in the practical class, just your mind is somewhere else and you are not developing this skill to make a good slide and to make sure that you have seen chromosomes with your eyes and you will be able to bring that again in your slide, you are wasting your time. You will not go very, very far off in your studies if you are actually want to develop a career in academics, right? So how many of you got a good slide in the class in mitosis? Especially mitosis I am talking. I told you the difference between mitosis and meiosis and how to pinpoint that in the microscope itself is this is. So yours was first year, but make sure that you develop yourself, grow yourself, and always connect theory with the practical. Correct? Okay with this? So with that I reach to the next slide. So let's come face to face. Right. So how are the things with you? You could follow? You could write all the new terms which I added here. Ma'am, can we see chromosomes during? Yes. Yes, see the how the chromosome, how will you see the chromosome? I tell you, it depends on what? First, good staining, right? Then good tapping, because that will separate the chromosomes, right? Cells and chromosomes also, meaning chromosome also and cell also, you have to have a good tapping, right? And then you will be able to see, all of you make sure that you have seen the chromosomes in the mitosis and meiosis both. Do it again, focus it again, take a picture with your mobile, but you are able to, we have good microscopes, so you should be able to see chromosomes in your slide. I would not say you will be able to see the crossing over or anything, but you should be able to see a chromosome in your slides, in your mitosis. Try, keep working, do it hard. You can find me wherever you are, you can send me pictures clicked by your mobile, but you are able to see the chromosomes, right? Stage is not much, we can see the chromosomes, but stage is not clear. I tell you, Sujal, what? Sujal, I tell you the answer to this question is what? What happens is, I told you it depends on these two things, how you have stained it and how you have stained it and how you have tapped it, right? If your staining is good and your tapping is good, what will happen? And were you there in the class where I explained you what is the difference in theory stages and practically what do you see? You were there, Sujal, in that class? Because there is a difference in theory and practical. Theory, we show you the exact point which we are talking about, but in practical, how the things appear practically. You were there, so keep those points in your mind and you can find me whenever you want to, but then you will be able to distinguish what is anaphase, what is metaphase, what is prophase in mitosis especially, because, but I tell you the trick is in the good staining, picking up good buds, good root tips, and then good staining, good tapping. You will get it, and it's just a trick and a skill. Where the skill lies, this is the skill which nobody will tell you, you will get that only in the classes, and you yourself have to make sure that now you know. Correct? You can repeat it, still we have 1 week to go before the exam. Friday again is there, Wednesday also you have a class, but make sure, because the time never comes back, maybe you really enjoy when you learn things thoroughly. Doing it just for the sake of doing does not take you anywhere, right, Sujal? And you see me in the class, I would love to, because you seem to be quite interactive and a good student to me. Okay, right? So let's go ahead, and where am I? Correct. So I told you till here, and then they are very, very important in evolution. I told you this also, and now I am here. Deletion, right? So deletion, I told you deletion also, so I don't expect the deletion. What I am telling you here, I told you already, is that deletion we studied during the complementation experiment, and deletion is used for, meaning advantages of these studies which we are talking about, the deletion very often is used for mapping and knowing the sequence of genes on a chromosome. And you know, this is chromosome banding, which you see here, is that different stains are used to understand the different regions of the chromosomes. So breakage, fusion, bridges, cycle, this I wanted for you to know, because once you know this, then you will actually understand the implication of small changes in the chromosome. That gives you huge changes, and it keeps on happening in the cell. So what is that? What is that? Please listen to me carefully. Is what I am showing you, that is the last slide. I tell you this also because I am almost done. Then I will talk to you about your questions. Is what is this? I am done. This is the last slide. So what is happening here is I am showing you this sperm, and what has it done is it has lost its telomere. What is telomere? Telomere is the last region of the chromosome. And what does it have? It has a heterochromatin, where the inactive form of DNA is present. And what is the role of this? Is that during mitosis, when the centromere is dividing, it allows the ends to remain separate, right? Like the one here. This end of this chromosome is broken by accident, and this end is not broken. This is normal. Now this chromosome, when it is undergoing mitosis, what will happen? It is broken, of course. So centromere will divide. One half will be pulled to one end, and the other will be pulled to the other end during anaphase. Okay, agreed with me? Now what will happen? Because this end was defective because of loss of this part, because this end was defective, so what will happen? It will not be able to separate. So when this opens up during anaphase, what will happen? This centromere is going to one pole, this centromere is going to the other pole, but it will still remain connected from the center, right? This was the bridge which you are seeing in the slides in your class, right? And this will go to the other end. Is this you have seen? Of course, you might not have understood, but now you understand how this bridge is formed, because the telomere of the parent chromosome was lost because of some accident. This is fine. And when sometimes a fragment is lost, that is laggards, because that does not have a centromere to pull, that keeps lying in the center, even though the rest of the chromosomes have moved to the other parts. But what is this implication of this chromosome bridge? What happens is now during anaphase, this chromosome will be pulled further, this centromere to the other part and this centromere to the other pole, and it will have pressure, it will break from anywhere, right? It will break from anywhere and it will form two new chromosomes, right, which have broken from anywhere. It might as well happen it breaks from here, it might as well happen it breaks from here, but it broke from 2 points, it's fine. But these chromosomes carry on with this defect, so they will again have this type of chromosome which centromere divides, and they will again give rise to this bridge. This is called as breakage fusion bridge cycle. This is a very important phenomenon which is leading to a lot of chromosomal aberrations and changes, because it breaks at any point in the next time. Could you understand this or not? The chromosome bridges and laggards you can understand is why, because it happens is that they have they need the centromere to be pulled. In laggards there is no centromere, and if it's not laggard, if the end is deleted, it will lead to the breakage fusion bridge cycle going on in the cell for generations unless they develop the final region. So how about this? Could you understand? I told you several terms. I can summarize quickly if you want to. Right? So let's do it this way. Write all the terms. Let's everyone start writing. I am starting with what? I started with what? Euploidy. Maybe it's sort of game. You can write the next term here. Euploidy के बाद what did I tell you? Right, I want an answer from all of you and then I will leave you. वाइवा के मार्क्स आप लोगों का तो मैंने पूरा काम कर दिया है. अभी मेरे को आपकी प्रेजेंटेशंस के मार्क्स लगाने हैं. 10 marks की प्रेजेंटेशन है. Yes, polyploidy. Then next, who is writing the next term which I told you? Yeah. लिखिए जल्दी जल्दी. मैं भी थक गई हूँ, काफ़ी देर हो गई. So I want to leave you now. But I want a quick recap or a summary from your side. Autopolyploidy, very good. Next one. There are so many students in the class. पायल राहा, लिखिए आप. अगला क्या बताया था? नेहा यादव, अंजलि, जो अच्छे से काम करते हैं, जो ध्यान से काम करते हैं, nobody can stop them from reaching to the top, I tell you. And those who pass time are not serious and they are discouraging others also, but they suffer. Yes, correct. Allopolyploidy, then amphipolyploidy, after that aneuploidy, after that. So you can keep on writing because it will take time, but I am looking for your answers. And I would like to stop this stream now. From my side, I have finished all your syllabus, but still I want to discuss one day the question paper if you want to. I have done my work and it took a lot of effort from my side, but still I want you to have a good foundation and a good knowledge level when you pass out from here. So if you are interested, I can discuss a theory question paper anytime, but that will be more for discussion like we discuss it here, that what to write and how to write in here. And anyone and anytime whenever you want to before your theory exam. When is your theory exam? Right, Koshambi. When is your theory exam? When is your theory exam? 3rd June, correct. So you'll have a lot of time before that, after the practical exam. Then if you feel like, you can request or you can suggest me, I need to look at that. But from my side, I've done everything and I'm looking for your presentations this Friday, and please make them correctly and don't waste my time also. But my agenda is just one, to develop you and take you wherever you want to go in your life. So I am just observing the students what they want to do, and I would really love to take some of you to the level of thesis in your 4th year. So that's my objective. So thank you very much, all of you, and I owe a big thanks from you because it took a lot of time from me, but I feel satisfied that I've done everything nicely. Okay, so I am the time to say bye bye to all of you, and I'll see you again on Fridays in Friday's class. Correct? Bye bye.